Hello everybody and welcome to today's talk in our Evolution and Ecology seminar series. Before I introduce today's speaker, a few short announcements. First, a reminder that there will be no talk on the coming Monday. Our next talk instead will be on Wednesday, and we Oded Javi giving his talk. And today, as usual, after the talk, there will be a Q&A session with our speaker. Please post your questions in the designated Slack channel, the link to which can be found in the description of the stream. And please upvote questions you would like to hear answered. And now it's my great pleasure to introduce today's speaker, Professor Nicole King. Nicole obtained her PhD from Harvard University, after which she took up a postdoctoral fellowship at the University of Wisconsin. In 2003, she moved to UC Berkeley, where she's a professor in the Department of Molecular and Cell Biology and an investigator in the Howard Hughes Medical Institute. Nicole's group uses a multidisciplinary approach to study the very fundamental and fascinating questions surrounding how multicellularity evolves using colony-forming choanoflagellates, which you'll tell us all about today. So today, Nicole's giving her talk with the title Choanoflagellates and the Origin of Animal Morphogenesis. Nicole, thanks again for accepting our invitation, and the floor is all yours. Thanks.
Thank you very much. It's a pleasure to be here. I was very excited when I saw that this seminar series was starting, and I'm truly flattered to have been invited to speak in it. Before I get started, I think I should make a disclaimer that I'm working from home, of course, and there may be a couple of distractions. There's Annie and Bunny, and they bark a lot. So my son's in charge of keeping them quiet, but sometimes it doesn't work as well. So we'll see. Okay, so I wanted to start out just by setting the foundation of what excites me about the research that we do in my laboratory. I'm fascinated by animal diversity, and I'm fascinated by the many different forms that animals can take on. But I think what's really exciting to me is this fact that all animals start in the same place. So they all start with a single fertilized egg, and that egg goes through multiple rounds of cleavage and to produce a simple and seemingly undifferentiated early stage embryo. And then after multiple rounds of cleavage, I went too far, this gives rise to the diversity of animal forms that you see here, as well as, untold further diversity. So it's fascinating to me to think about what are all of the different ways that you can get from that single cell to these beautiful multicellular forms. And of course, one of the ways in which we've made so much progress in terms of figuring out the mechanisms underlying the transition during development from a single cell to these beautiful multicellular animals is through the use of model organisms. And so a few examples here are, of course, worms, flies, zebrafish, and mice. And by studying these organisms in molecular and cellular detail, we've been able to reconstruct shared molecular and cellular mechanisms that we can infer are also in place in some of these non-model organisms that we find equally fascinating. Now, I've become interested in a similar transition, but this is not over the life history of a single species, but instead over macroevolutionary timescales. And that is, how can we understand the evolution of animal diversity and animal multicellularity from their single-celled ancestors?
Sorry, Nicole.
Yeah, sure.
We're still seeing your title slide.
You are. Okay, hold on. That's why I was concerned. On my slide, it says resume share and says it's paused, and I don't know how that happened. I'm going to stop the share and restart it. Sorry. I'm having a problem. After all that. We had this working, everyone, before we got started. Okay, here we go. Is this working now? Let's see. Can you see it moving now?
Yep.
Okay, thanks
Okay.
for letting me know. Okay, so I made a beautiful series of comparisons that you all missed, saying that, you know, we can understand how you go from a single cell to a multicellular form over development, and that we can learn about the diversity of these, how these diverse forms develop by comparing the underlying molecular and cellular mechanisms to what we see in model organisms. And so here what I'm showing you is that we can use a similar mindset to try to reconstruct over macroevolutionary timescales how animals evolved from their single-celled ancestors. And a key thing that we need to keep in mind here is that animals are monophyletic. They all evolved from a single common ancestor. And so when we compare the biology of diverse animals, we can start to reconstruct features of their last common ancestor. But what I'm interested in, and what my laboratory is interested in, is trying to understand the processes by which that first animal evolved. And in particular, we're interested in the evolution of multicellularity from what were presumably unicellular microbial eukaryotes that were ancestral to the animal stem lineage. And then we want to understand mechanisms underlying the evolution of cell differentiation and coordinated function. So some of the questions, the big questions that motivate us are, first of all, we want to understand what was the nature of the first animal. Can we understand with greater precision and detail what that first animal looked like and how it interacted with its environment? Can we understand the mechanisms underlying the transition, the evolutionary transition to multicellularity, and can we start to understand it with the same level of mechanistic detail that we now understand how development itself works? Can we use an evolutionary mindset to reconstruct the ancestral and potentially core functions of animal genes, and use these evolutionary types of comparisons to perhaps uncover functions that are difficult to really access in a pleiotropic environment? And then we've unexpectedly become interested in how bacteria might have influenced early animal biology. And I'll talk about that very briefly. So to do this work, obviously, we can't go back in the fossil record. First of all, the fossil record does not provide a clear representative of the first animals and their ancestors. But secondly, the fossil record wouldn't give us access to molecular and cellular mechanism. So instead, we focus on the closest living relatives of the animals, the choanoflagellates, which I will sometimes call the choanos. So the choanoflagellates are the closest relatives of animals shown in this simple tree. And they're, in fact, as diverse as animals in terms of their genetics. And I'll come back to that very briefly later. Now, the idea then is that if you use a phylogenetic framework, then features that are shared among all animals, we infer were also present in their last common ancestor, and features that are shared between animals and choanoflagellates, then we can infer were present in their last common ancestor, which we're calling the Ur-Choanozoan. And so for my talk today, what I'd like to do is give a brief history of choanoflagellate research to give a sense of the trajectory of how choanoflagellates sort of entered the molecular and cellular era. I'll then give a personal narrative about how we've established choanoflagellates as experimentally tractable models that we hope anyone could study in their lab if it helped to address questions that interested in them. And then I'll close with what has been a surprising story for me, and that is that we began to do field work, you know, frankly, just for fun, and unexpectedly it led us to exactly the types of molecular mechanisms that we were hoping to uncover through our laboratory work. And so those are the main points that I'm going to go over in this talk. So I'm going to start by introducing you to choanoflagellates. I'm never sure how familiar people are with them, so bear with me. So choanoflagellates are a group of microbial eukaryotes, and they have sort of a diagnostic cellular architecture, which is this 5 to 10 micron cell body, a long apical flagellum, and a collar of actin-filled microvilli. And the name for choanoflagellate comes from choano, which is for the collar itself. And this is a feature that is universal within the choanoflagellates. Now, the way these cells work is that the the flagellum undulates back and forth, and this creates water currents that pull material out of the water column, in particular detritus and bacteria. And choanoflagellates, in fact, make a living by eating bacteria. And so you can see in this SEM a bacterial prey that's been captured against the outside of the collar. Now, today we can easily recognize choanoflagellates under the microscope, but early microscopy obviously didn't have the same level of resolution. And so it's really, to me, fun and interesting to look at the prehistory of choanoflagellate research. And this is something that's really been tracked down by Barry Leadbeater and summarized in his book, The Choanoflagellates. And so you can see that the first plausible choanoflagellates were documented in about 1786 by Mueller. And here by Ehrenberg in 1831, we're starting to see these potential trajectories, or maybe these are flagella. Here's a replica of this in greater detail. But it's not until 1849, and then in James Clark's monograph in 1867, that we start to see cells that are indisputably choanoflagellates. And even here, you can see that James Clark was documenting the collar not as microvilli with distinct actin-filled microvilli, but instead as a continuous funnel. But what I think really launched choanoflagellates into a more important space was work by people like William Seville Kent, in which he documented that the choanoflagellate cell architecture was nearly indistinguishable from a group of cells found in sponges called choanocytes, or collar cells. And just like the choanoflagellate, these collar cells had an apical collar that we now know is also made of microvilli, and a long apical flagellum. And as well, just like choanoflagellates, the collar cells of sponges have an important role in capturing bacteria, which is the major food source for sponges. Pardon me, I'm just gonna have a sip of coffee. So right about this time, Haeckel was starting to think seriously about how animals might have evolved. And one of his proposals was that the ancestors of animals might have been colonial flagellates. And these are not actually Haeckel's original drawings, these are reproductions. But the idea was that there might have been an undifferentiated colonial flagellate, now here depicted as a colonial choanoflagellate, but Haeckel wasn't that specific. And the idea is that animals, because all animals pass through this early undifferentiated blastia stage, that might have been, that something that approximated that might have been on the stem lineage to animals. And that later cells within this colonial form might have differentiated to produce gametes. So this is the blastia hypothesis, and a corollary to that was the gastria hypothesis, in which there might have been some infolding within this blastia that would lead to an early precursor to gastrulation. So I want you to keep this in mind, this idea of this in-pocketing, because we'll come back to it later. Now, Haeckel, it turned out, and Saville Kent, who I mentioned previously in the context of comparing choanoflagellates to choanocytes, actually really did not get along. They had competing views of how animals might have first evolved. And one potential challenge to this blastia and gastria hypothesis was the discovery by Saville Kent of this enigmatic organism, which he called Proterospongia haeckelii. In fact, this was a dig at Haeckel. He named this organism after Haeckel because he thought that it undermined the blastia hypothesis. And the reason is that this multicellular thing that has clear collar cells embedded in some sort of matrix, he observed to have amoeboid cells in the middle. So that the idea is that it had differentiated cell types, and that these were in the center, not through gastrulation. The problem with this is that nobody has ever documented this organism since then. Um, and so it is unclear whether Salvokint actually saw this organism, or whether he might have elaborated on some wishful thinking about what might exist out there. I won't have time to come back to it in this talk, but we do have very exciting new results of choanoflagellates that actually can transition into this amoeboid form. And so this, I think, has made us increasingly interested in the relative support of these two different hypotheses. Okay, I want to move forward then on the trajectory of this of the study of choanoflagellates, and say that now in the 20th century, or not now in the 20th century, in the 20th century, that being the last one, there was increasing interest in documenting choanoflagellate diversity. And in fact, although I came to the study of choanoflagellates not as a protostologist, but instead as someone who had trained in genetics and biochemistry, I thought of choanoflagellates as a pretty monomorphic and boring group of organisms. In fact, they have a tremendous amount of diversity, both in their morphology and their ecology. And here you're just seeing a small sampling of some of these incredibly diverse and beautiful forms. One of the things that really inspired me to study choanoflagellates was this 1983 paper in which Barry Leadbeater, shown here, documented life history, a simple life history of a choanoflagellate that he isolated from nature. And what you can see here is that he observed that this organism could switch from a sedentary stage into a swimming stage. It could also form these sheets of cells in a multicellular form, and it could convert into this minute cell type. So here was a seemingly simple choanoflagellate, but in fact it had multiple stages to its life history, and it could differentiate between different life history stages. About 10 years later, a group at the MBL started building phylogenies that were largely focused on understanding the relationship of animals to other major groups, such as fungi, but they were the first, to my knowledge, that included sequences from choanoflagellates. And so this is a tree based on 18S sequences from 2 different choanoflagellate species. Interestingly enough, there was no real support for this branch, but these were grouped together, and this alone was enough for me to get really excited about this. So there had already been these early comparisons between the cell morphology of choanoflagellates and sponge choanocytes, and this moved it into the molecular era. And so for those reasons then, I decided to try to study choanoflagellates as a model for reconstructing the origin of animals. And so that's what I've been trying to do now since I started in 2000 studying choanoflagellates. Okay, so what I want to do next is really focus on some of the work that we've been doing in my laboratory to, pardon me, bring choanoflagellates into the laboratory and make them useful for trying to reconstruct molecular and cellular mechanisms. And this has been a really fun process. It started with trying to, what I'm calling, domesticate a species, make it amenable to the types of molecular and cellular approaches that I had learned in the study of flies and bacteria. We then found that that there in fact was a great deal of interesting life history transition, and so we needed to gain experimental control over these. We've sequenced the genomes of choanoflagellates, and we've established forward genetics and genome editing. And I'm just gonna go through these quickly to give you a taste of what it's like to start a model organism from scratch. So the star of our story here is this species, S. rosetta. And S. rosetta is, let's see, S. rosetta was isolated from nature in this multicellular form. And this was exciting because if you remember back, early embryogenesis in animals typically goes through a blastia stage in which there are seemingly undifferentiated cells in a ball. And so here, this choanoflagellate from nature was actually forming a similar structure. And this harkened back to Haeckel's hypotheses about the blastia and gastria stages of animal evolution. And so I wanted to study this organism in great detail. Now, not only does this choanoflagellate form these balls of cells that look like a blastia, but in fact, there are aspects of the way it formed that I think make it additionally relevant to animal development. And that is that instead of the cells coming together through aggregation, akin to the way that the slime mold dictyostelium becomes multicellular, this choanoflagellate becomes multicellular through serial rounds of cell division. So all the cells are clonal. And so you can see over here a group of stills from different stages of this developmental process in which a single cell undergoes serial rounds of division to become this multicellular choanoflagellate. And you can see that in this video from work by Steve Fairclough when he was in the lab. You can see this single cell dividing over and over to produce this multicellular three-dimensional form. So that alone was fascinating. This choanoflagellate was undergoing a transition from being single-celled to multicellular in its life history. But the more we studied the choanoflagellate, the more we realized that it could undergo really interesting life history transitions that were similar to that of the species that had been studied by Barry Leadbeater, although it was not the same species. So this is the unicellular form that I just showed you, and it can transition into this multicellular rosette, but it can also become these linear chains of cells also through cell division in a different process. This part of the life history, the cells are propagating through asexual reproduction, but the choanoflagellate we discovered also has a sexual phase in which it can convert or differentiate into gametes. These gametes are sometimes differentiated morphologically. They can fuse. And then the diploid stage we think is the sedentary form, which can then re-enter the asexual portion of the life cycle. And so this was all fascinating to us in the context of understanding the evolution of cell differentiation. But even more fascinating was that we found that some of these life history transitions could be induced and were sometimes required cues from environmental bacteria. And so early on, we discovered that the switch from a single cell to multicellular form required cues from environmental bacteria. And the first species that we found that could induce this transition was from this genus Algorophagus, which is one of the Bacteroidetes bacteria. I just shared that for the microbiologists among you. An entirely different genus of bacteria, Vibrio, related both to, well, this one was actually Vibrio fischeri, the bacterium that regulates light organ development in the squid, Euprymna. This organism produces a completely different cue that can induce this switch from asexual reproduction to sexual reproduction. And so we're fascinated moving forward to understand whether bacteria might also be regulating other parts of the life history, or if we might be missing additional portions of the life history because the correct bacterial cues are not available. And that's something that we're continuing to explore in the laboratory. Now, I've sort of told this, I have to let the dog in, hold on. Come on, oh, you have Renee? Sorry, the reality of having a puppy. Okay. So I told this out of order because actually one of the first things we did in studying choanoflagellates was to sequence their genome. And that's because that was something that we could do without having all of the other experimental portions of our work done. And it also helped us to immediately start to address the evolution of animal genomes. And so I won't go into all the details, but simply to summarize that through sequencing the genomes of 2 different choanoflagellates, and now the transcriptomes of an additional 20 choanoflagellates, we've been able to gain increasing resolution on the evolution of animal genomes. And so what we found is that we have a much stronger handle on those types of genes that are likely to have evolved on the animal stem lineage. So here, the ones that truly are unique to animals and might have specific roles in regulating animal development. And so some of these are genes that have been long studied by animal developmental biologists like Wnt and Frizzled and beta-catenin, classical cadherins and hedgehogs, and a variety of different transcription factors. But I think what was equally exciting was the fact that many of the genes that had been studied in the context of animal development and animal cell biology are in fact found in diverse choanoflagellates. And so what that tells us is that these genes actually evolved before the diversification of the choanoflagellate and animal lineages, and they may have functions at a unicellular level that have not been straightforward to identify in the multicellular context of animals. And so these include C-type lectins. Although choanoflagellates don't have classical cadherins, they have diverse other cadherins. We've become very excited about genes like G protein coupled receptors that are involved in, for instance, olfaction in animals. And then interestingly, we've found a number of genes that are homologous to genes required for immunity, innate immunity in animals. So there's a lot to unpack here. But I think one of the things we would be really excited about is starting to reverse engineer some of these animal-specific genes into choanoflagellates and seeing if they can help us to understand pathways by which multicellularity and development might have evolved in this stem lineage. Now, in the study of flies and worms and other model animals, one of the key things for linking gene function to morphogenesis, of course, has been forward genetics. And so we also aspired to establish that in choanoflagellates. And I have to say, this is something that I didn't think could be done in choanoflagellates. I won't go into all the reasons why, but there were some important technical challenges in choanos that don't exist for model animals. And so I was pessimistic about this being something that we should tackle early on. But a very brave graduate student, Tara Levin, thought that it could be done, and she forged ahead and she succeeded, which was very exciting. And so here what you're seeing is just a panel of some of these classes of mutants. So here on the left is wild type, and you can see here in the absence of inducing factors, it forms these multicellular chains, and it can also be single-celled, but in the presence of the inducing factors that I discussed before from bacteria, it can actually be induced to switch into this multicellular rosette type form. And then what Tara did was she mutagenized this strain, and through that process, and then looked at individual strains of mutagenized choanoflagellates. And what she was able to uncover is a diversity of phenotypes, mutant phenotypes, that she grouped into different classes. So this one is what we call the rosetteless phenotype, because in the uninduced form, it's perfectly normal, but when it's induced, it never forms rosettes. So here you can see another chain. And you can see there's a lot of diversity. There's one form that's clumpy in the uninduced form, and it's also clumpy in the induced form. And so Tara went forward and focused on this strain to see whether she could figure out what genes were underlying this mutant phenotype. So there's Tara. And what she discovered is that the first one that she mapped was actually mapping to a gene called a C-type lectin. And this is a gene family that's also found in animals, where it also regulates development and immunity. And when she localized this protein, it actually localizes to the basal pole of the cells in the colonies. And we think that it localizes to the extracellular matrix and helps hold the colonies together. Now, another graduate student, Laura Wetzel, focused on this class, this very interesting class of mutants, where they are clumpy in the uninduced form and in the induced form. And she found that two different mutants mapped to a different group of genes called glycosyl transferases. And these are enzymes that regulate the structure of the extracellular matrix. And what we think is that, in fact, by disrupting these glycosyl transferases, she had disrupted the extracellular matrix, now allowing cells to stick together through aggregation. And that aggregation prevented the cells from developing into rosettes. And so through this type of work, we can start to map out which genes are required for proper development in the choanoflagellate. And again, in the case of these glycosyl transferases, they also play important roles in regulating development in animals. Now, finally, I think one of the very exciting advances in molecular cell biology has been the ability to edit the genomes of model organisms. And this is something that, of course, we wanted to do in choanoflagellates, but again, we faced a tremendous number of technical hurdles, and these were all overcome by David Booth, who was a really terrific postdoc in the lab and has started his own lab now. And I'm just showing you one example here. This is the mutation in the C-type lectin, this is the mutation that generated this phenotype here. And what he was able to do is develop genome editing from scratch. This is a multi-year process, very, very challenging. And here I'm summarizing this all in one slide just to show you that as an example, he generated a mutation here through CRISPR-Cas9 genome editing. And this resulted in the same phenotype as the mutation that is generated in this forward genetic screen. And so you can see here, this is the original mutant with this chain that's produced. And now here's the mutant engineered in by David Booth, and we get the same phenotype. And so we now think we're on the cusp of being able to walk through the genome and disrupt genes of interest at will, which will allow us to start teasing apart ancestral functions of genes that are shared with animals. Now, as an aside, I want to say that both Tara and David are starting their own labs right now in the midst of this pandemic, but they are both fantastic mentors, and I know that they're excited to welcome people into their labs. So if you find this work interesting, I encourage you to reach out to them. And because of their work now, choanoflagellates truly are experimentally tractable. So the kinds of things that you can do with cell lines, we think you can now also do with choanoflagellates, but choanoflagellates are living in their natural state. They're untransformed in the laboratory. And so they are really wonderful models, not only for reconstructing animal origins, but also looking at interesting cellular and molecular mechanisms involved in host microbe interactions, core functions of proteins and protein families, morphogenesis, the regulation of cell differentiation, et cetera. And so we're very excited about this as an organism moving forward. Okay, so in the last period of time, what I want to do is switch bases. I mean, what I just told you about was this very stepwise and focused long-term effort to try to drag choanoflagellates out of a descriptive world into a world in which we can really tease apart molecular mechanisms and use choanoflagellates as experimental models. But what I want to do now is switch and tell you about how doing field work somewhat on a lark led us to molecular and cellular mechanisms that we never would have known to look for in the first place. And this all starts on the island of Curacao, which I hope I will eventually get back to. Because it was really wonderful going there and learning how to be a field biologist. And I should say that the only field work that I had ever done prior to this was as an undergraduate student at Indiana University, learning how to do field work with birds. I was part of a research group well, let me fast forward and say that for the last decade or more, I've been part of a really wonderful research group that was initially organized by Patrick Keeling through the Canadian Institute for Advanced Research. And one of the great things about this research group is that we would all come together once a year or twice a year to discuss our latest. And we really got to know each other. And I was greatly influenced by the other researchers in this group, many of whom were microbial ecologists or people who were studying the biology of microbial eukaryotes in the field. And so they really helped me think about choanoflagellates not simply as bags of genes or points on a phylogenetic tree, but really think about them as organisms. And so at one point they started saying, rather than, meeting in a conference room and showing slides as I'm doing for you right now, how about we all come together and try to do some science together and really influence each other's science? And so they convinced me to travel to Curacao along with a few members of my lab and see what we could do as field biologists. And so this is where we were. It's called the Carmabi. It's a marine station in Curacao, and it's this yellow building in the back here. In front of it is a beach. This is the view out over the ocean from the marine station. And the laboratory, was not highly equipped. So we were in this room with a folding table and a microscope that we brought. And this is my son as my laboratory assistant. And the first time we went there, my goal was simply to figure out what this whole fieldwork thing was about and to try to figure out if there was anything we could do that would actually make a contribution. So the first time I went there, I brought one graduate student, Ben Larson, and we went around the island collecting choanoflagellates, and it was really fun. But I wasn't sure what we could learn and really contribute. But we decided we went back a second time, again with this group of researchers led by Patrick Keeling. And this time, in addition to Ben Larson, shown here, heading out into the water to collect some water samples, Tess Linden, a graduate student, and Thibaut Brunet also came. And the three of them were an amazing team. And everything I'm going to tell you was the work of the three of them working together. So on this trip, we decided let's really have a goal of making some sort of scientific contribution at the end of this. And we thought, at a minimum, why don't we describe the diversity of choanoflagellates in Curacao? We had this picture of, we're going to write this paper. It's going to be called Choanoflagellates of Curacao. And it would be our first sort of natural history type of study. And so that's what we were doing. We were going around to lots of different environments. We were learning how to, to take GPS coordinates and measure salinity and do those kinds of things, which are probably second nature to many of you, but were new to all of us. And we were learning a lot in the process, and we hoped that we would have a whole new array of choanoflagellate diversity to study in the lab. But that all was thrown by the wayside after a trip by Ben, Thibaut, and Tess to this unassuming splash pool. So these splash pools, this is the windward side of the island. You might be able to see windmills in the background there. And this side of the island is being constantly bombarded by wind and waves. And this surface here is actually old dead coral reef. It's really jagged and hard. This is a pretty harsh environment. It's hot, it's humid, it's windy. There's nothing really living. Well, there's nothing macroscopic living out there. And there are these occasional splash pools, and you can see one of them here. And so Thibaut took a water sample out of this, and they brought it back to the lab at Carmabi, and they put it on the microscope, and they could not believe what they saw. And I still remember the first time I saw this video. So I'm going to share this with you because it just blew my mind. So here what you're seeing is a multicellular choanoflagellate. They weren't 100% sure that's what they were seeing at first, but now that's been confirmed. And hopefully you can see this haze on the outside. And these are all the collars and flagella pointing outward. And what I want you to do is ignore these predatory dinoflagellates. We don't care about those. We're going to focus on the choanoflagellate. So what you're seeing is a multicellular choanoflagellate, and it's cupped, And you're looking at the lip of the cup to start, okay? So I'm gonna start this movie, and so you can see what they saw. Whoa, it's amazing. So this thing sort of flattens out and then completely inverts. And then here's another one of these multicellular choanoflagellates. And now it reverts back to its original state now, you see, with the flagella on the outside. So it can entirely flip its curvature inside out. Now it can swim away. This one will flip again. So now watch those collars. They're flipping from the outside. And now they're all on the inside. And so this is amazing. This is multicellular behavior in a choanoflagellate. And it was tremendous. And so all goals of trying to describe all the diversity of choanoflagellates on the island of Curacao went by the wayside, and we decided just to focus on this choanoflagellate. So this was exciting because it was reminiscent of the type of epithelial tissue bending that you see in animal development. And so a few examples, for instance, gastrulation that's been well documented in the fly, and neurulation in the chick. And it turns out that this tissue bending is nearly universal in animals. And so it's also found during development in a sponge. You can see here that you can go from this stage to an infolding of this cell layer. In the sponge, we see it in Nematostella, which is a cnidarian. Again, you see this gastrulation process. And ctenophores as well seem to have this tissue bending. So this is just mind-blowing and very exciting to see this in choanoflagellates. And we wanted to know whether the mechanisms of tissue bending in choanoflagellates were similar to those that are seemingly conserved across animal diversity. And so what Tess, Thibaut, and Ben started out by doing is trying to figure out, first of all, what regulates inversion, how do these sheets invert, and how did this evolve? And again, remember, I mean, I think to me, one of the exciting things about this project was just how quickly it went. Understanding the answers to these types of questions in model animals has taken quite a long time. I think we have an excellent understanding now. But by using these evolutionary comparisons, they made great progress very quickly, despite the fact that this organism, this is a new species. I forgot to tell you this, we've named this species C. flexa for the fact that it undergoes this flexing behavior. And we had no genome sequence at the start. There were no molecular tools because this is something we dug out of a pool in Curacao, and it hadn't been studied before. Okay, so the first thing is what regulates this inversion. And this was a little bit tricky at first because although we could see the inversion happening seemingly spontaneously in the lab, it wasn't obvious to us what actually triggered it. But that changed when we brought one of these cultures to the MBL in Woods Hole and let a lot of the students there start studying it. And what they found was that the more they looked at these cultures, the less they would invert. So they would look at them for hours, and and the cultures would essentially stop inverting. And so the TAs in the class and I were confused, and we were trying to figure, it was very frustrating because we wanted them to study this inversion process, but the colonies weren't inverting. And then we realized, oh, they've been sitting on the microscope this whole time. Maybe light has something to do with it. And in fact, that proved to be the answer. So what you're seeing here is a low magnification view of one of these cultures. And each of these shiny blobs is a C. flexa colony. And what we found was that if we put them under the light like this, and then we turned the light off, you'll see what happens. So here's a movie, and now the light has been turned off. And now you can see that in response to the light being turned off, the vast majority of these colonies are balling up and starting to swim around. And that balling up is the result of this inversion. And we also then developed a way to quantify this inversion process. And so you can see that here. What we do is we measure the sheet area, and then we normalize it to 1 at the start of this experiment with the light on. And then when the light is turned off, so we're essentially measuring sheet area across the population and normalizing it. And then when the light is turned off, you actually see this initial expansion, and then the sheet area drops. And so what's happening is that the sheet flattens out and then it inverts. Hopefully you can see that. Okay, and so this provided a quantitative way to do the types of mechanistic experiments that we wanted to do to really understand this. So the next thing we wanted to do was figure out what is the photoreceptor. In animals, our ability to respond to light is dependent upon photoreceptors. Choanoflagellates were not previously known to have any sort of light response. And so we wanted to figure out how were they sensing the light and how was that getting translated into this morphogenetic process. And again, normally it would have been extremely difficult without genetics or some other type of mechanistic approach to figure out what the photoreceptor was, but we were lucky because a group of scientists in the field of optogenetics had been scanning through genomes of choanoflagellates, and they had already found a gene that proved to be a good candidate. And so this is the gene that they characterized. This is one of the publications on this protein. And it's what's called a Rho PDE, short for rhodopsin. So there's a rhodopsin portion of the protein and a phosphodiesterase portion of the protein. The rhodopsin responds to light when it's in the presence of retinal. And they showed that this rhodopsin can result in the activation of this phosphodiesterase domain. Now, the phosphodiesterase is an enzyme, and it converts cyclic GMP to 5' GMP. Now, cyclic GMP is involved in signaling cascades, and so this conversion then inactivates this. And so without knowing anything else about the choanoflagellate, Thibault, Tess, and Ben hypothesized that this might be the photoreceptor. And there were a lot of, if it was the photoreceptor, that would have been a lucky break because it allowed them to make some really exciting predictions that they could test. So if this is the photoreceptor, then depleting the cells of retinal should lead to inversion. Inhibiting this phosphodiesterase should also lead to inversion, or flooding the cells with cyclic GMP should also lead to inversion. And so they did all 3 of those experiments. I'm just going to tell you about one of them. And that is this experiment in which they found that strain lacking retinal is blind. So in the presence of normal environmental bacteria, what you see is that when the lights get turned off, the colonies flip. But when they killed all the environmental bacteria and replaced only with a bacterium that lacks the pathway for retinal, this P. oceanii, you see that it doesn't flip. And this is also, and this can be reversed when they add back the environmental bacteria here in blue. And so this suggested, that at least whatever the factor is, it's coming from bacteria. Was it in fact retinal? Well, if they take that, so here again is this negative control with the strain that doesn't make retinal. If they now add retinal back to it, you can see that that restores light perception. And so I'm going to skip ahead on this and just say that all of these predictions proved to be correct. And so this is, we think, the very best candidate for the photoreceptor. And so it's exciting, this brand new organism that has this really cool light sensing behavior, and they were already able to figure out how this organism is perceiving light. The next question was, how do sheets invert? And this was very exciting because it turned out to be related to the ways in which animals do tissue building. So one piece of background information you need is that the cells in this multicellular form are actually held together by by their microvilli in the collar. And so you can see them align, these collars aligning to each other here. And then in TEM, you can see that the microvilli are paired up, whoops, where cells are adjacent to each other. And we can also see that the collar changes shape during inversion. So here's a choanoflagellate colony in which all of the flagella are in. And hopefully you can see that these collars are parallel, whoops, in a sort of a barrel shape. When they're induced to invert so that the flagella are on the outside, now the collars open up. I have to stand back so you can see me. Wait, there we go. And so the collars opening up pushes the curvature of the colony outward. How is that actually mediated at the level of the cell? We know the light, that they're sensing light, but how does that get translated into changes in cell morphology? I'm going to skip over a lot, but simply say that drug treatment with various inhibitors of actomyosin all block this inversion process. So this implicates actomyosin in the inversion process. And in fact, when Thibaut Brunet raised antibodies against choanoflagellate myosin here, and also stained with a stain for actin, what he found was that at the apical poles of all the cells in colonies, there are rings of myosin, and these are surrounded by F-actin. And so, again, skipping over experiments that I don't have time to show you, he was able to demonstrate that inducing the flipping or inversion behavior results in contraction of the ring. And so there seems to be that myosin is involved in regulating this morphogenetic process at the level of single cells. And so what this looks like is that light activates the rhodopsin, and this activates the phosphodiesterase. And in this state, in the light, the cells have this curved collar with all the flagella on the inside. And in other work that I'm not showing you, this is the feeding state. Whoops. When the light is turned off, this inactivates the rhodopsin, allowing cyclic GMP to accumulate. And this results in contraction of the myosin ring and results in a change in the collar morphology so that it opens, flipping the curvature of the collar and switching it into this swimming state. Is this true just for C. flexa? Well, in fact, Thibaut found this contraction exists in lots of other choanoflagellates. I'm showing you one example here. And if you map this contractility onto a phylogeny, here you're seeing that all animals have an apical actomyosin ring and engage in individual apical contraction. And now Thibaut has found that all of the choanoflagellates he's looked at also have an apical actomyosin ring, and they all engage in apical contraction. And so we infer then that both of these features were present in the last common ancestor of choanoflagellates in animals. Now, one difference is that animals do collective apical contractility, for instance, during gastrulation. And we also see this in C. flexa, as I just showed you. What I didn't show you is it also exists in a close relative called C. perplexa, but does not seem to exist in these other species. And so for now, we think the most reasonable inference is that this ancestral module for apical contractility was co-opted at least twice to mediate collective contractility. And so through this and lots of other work, we're starting to build a picture of the last common ancestor of animals, which was a simple multicellular form that had collar cells and 8 bacteria, and probably had some form of apical contractility that allowed it to undergo tissue bending. And then in work that I also didn't have time to talk about today, we think that it reproduced sexually, producing eggs and sperm that fertilized to produce a zygote. So just as we today all developed from a single cell, we think that that was also true for the first animal. And so I will close by thanking all the members of our laboratory and one of our more recent photos, and I'm excited to take any questions. Thank you. Should I stop sharing?
Yes, please. I think that would be handy.
Great.
Thanks very much. Yeah, fascinating stuff.
Thank you.
We will have time for some questions. People are still probably submitting them and thinking about them.
Sure.
So in the meantime, can you speculate a bit about what's so special about Perplexa and Flexa, or what the potential reason of why they evolved this flipping potential?
So, so essentially, there are these. There's the one mode of multicellularity I told you about first, which is this cells dividing and staying together. In that form, the cells are often sticking together. A part of the mechanism is through incomplete cytokinesis, so the cells are physically attached. And so that the physical attachment, I think, limits, and they also use a central extracellular matrix that limits the ability of the cells to move relative to each other. In C. perplexa and C. flexa, the cells are attached through their collars, and that gives them some flexibility. So as the collar changes its shape, the cell bodies can move relative to each other. And so I think that's an important part of it. I should say that, I mentioned earlier on that choanoflagellates have cadherins, and we don't yet know what mediates the interactions between the cells. But the only time, when we've stained for cadherins before, they're actually in the collars. And so it's possible that the same mechanisms that mediate cell adhesion in animals might be mediating those interactions between collars and choanoflagellates.
Do you think the adaptive function of that is related to feeding? And could it be that there's more, in these pools, potentially there's more stagnant water, so there's higher densities of bacteria that they can trap inside this?
Yeah. I tend to think of this, so the default state, if you leave them alone, is the feeding form. And the feeding form is when they have their flagella facing in. They flip to this swimming form when the lights are turned off. It turns out that they flip in response to a lot of stressors, like mechanical stimulation will get them to turn. We've also been playing with various inhibitors, and that gets them to flip. And so I think of the flipping as actually an escape mechanism. And once the escape has happened, they go back to feeding.
Okay, we've got a couple of questions or a few actually.
Okay.
But there's one question: are some of the genes that are inherited from the urchromosome also found in other protists or in fungi?
Yes. So, when I started studying choanoflagellates, it was thought that, the textbook version of animal genomes was that they had cadherins and receptor tyrosine kinases and collagens, and these were sort of definitive, they defined animal genomes and animal biology. We now know that many of these genes exist not only in choanoflagellates, but also in other relatives such as Capsospora, Ministeria, these other microbial holozoans that are closely related to animals. Certainly, nothing is completely new in any extant lineage. So there are certainly a lot of genes shared with other eukaryotes, including fungi and slime molds and plants. Many of those genes are essential for basic housekeeping functions. And so I think what's challenging and what is exciting is figuring out which of those genes actually contributed to the evolution of multicellularity in animals. And so that's what we're trying to figure out.
Cool. There's one person burning to know more about the amoeboid cells. Could you tell?
Yeah, I wish I had more time to talk about those. So this is the work of Thibaut Brunet, and we're actually writing up this work right now. And again, we've been so lucky with lucky observations. So he was studying S. rosetta in the laboratory, and he had a culture with a lot of bacterial biofilm in it. And he was shocked. He was looking into the microscope, and he was shocked to see them convert from a flagellated form to an amoeboid form. And he's actually, his whole focus is studying the evolution of cell contractility. So this was right up his alley, but we didn't think that choanoflagellates could form amoeboid cells. So initially he thought, well, maybe it's induced by these bacterial biofilms, because we'd seen that type of interaction before, but it turned out it had nothing to do with bacterial cues. What it had to do with was that the cells were being compressed. And this is a common inducer of amoeboid cells in animals and also in chytrid fungi. And so he's been studying this in more detail. And sure enough, under compression, choanoflagellates consistently, and again, diverse choanoflagellates, not just S. rosetta, convert into this amoeboid form. And again, there seem to be shared molecular mechanisms with animals. So it's a thrilling, I can't tell you how excited we are. I wish I'd had more time to talk about it. And I think it really does force us to, as does the work of Iñaki Ruiz Trio, where he's looking at amoeboid cells as well, force us to really think about the diverse cellular morphologies and cellular modules that might have been in place in the last common ancestor of animals.
Yeah, very exciting, yeah.
Yeah.
Unfortunately, I think we'll have to leave it here. There's still plenty of questions in the Slack channel.
Yes.
If you wouldn't mind, and if you are on Slack, maybe you could go through them and answer some more if you're interested.
I'm happy to, and I'm sorry that I ran over.
No, that's okay. Maybe
this was really fun.
Maybe I wasn't quite enforcing enough, but it's fine. It was a fascinating talk. Thanks again very much. And as I said, there's loads of more interesting questions.
I'm happy. I'll sit here and answer them.
Brilliant. All right.
Okay.
So with that, we're at the end of today's talk. Just a reminder, no talk on Monday. Our next talk is on Wednesday. Oded Rehavi from the University of Tel Aviv will be talking about how C. elegans parental experiences regulate a critical decision made by the progeny. We hope you can join us again. You can stay up to date about upcoming talks by joining our Slack channel and by following Evo Eco Seminars on Twitter. Thanks for watching and have a great weekend.